Welcome to my presentation, but also welcome to the new era of scientific publishing, because I believe you've made the first step into such an era. This is the anniversary of iOS Press, and press comes from printing press, but that's not how we publish scientific findings anymore, of course. We do it digitally. We have a sort of scientific knowledge cloud that we access through the internet. Researchers upload their findings there, and whoever is interested, at least in the open access case, can access this cloud and get the insights, the scientific knowledge they want. So I think this scientific knowledge cloud has been a huge success, but there are also very serious shortcomings. Imagine this simple task for a developer. Let's say a developer who wants to build an application that somehow uses that scientific knowledge. That task could go as follows: retrieve all genes that have been found to play a role in a part of the respiratory system in COVID-19 patients, and moreover only include results from randomized control trials from the last 7 days. So to a naive developer who doesn't know how we publish scientific findings, this might sound quite easy, right? I just have to find the right API, formulate the query, maybe do some post-processing and filtering. But of course, we all know this is much more complicated, because scientific results are represented in plain text. So we have an abstract in text, and the interpretation of that text is not immediately accessible to algorithms. So we need text mining. Well, 5 years ago, there was the 30th anniversary of iOS Press, also here, and I was also giving a talk. And I argued back then that text mining is not good enough. And I pointed to a workshop that's called Biography that took place in 2015. And in these kind of workshops, the world-leading research groups come together to present their best text mining systems and to compete in concrete tasks. So in this case, there was a task on relation extraction. So to extract from scientific abstract, for example, that a certain thing A has a certain relation to a thing B. And here A were chemicals and B were diseases in this specific case. And there the F-score of the best system was just below 60%. And I argued back then, this is not good enough. But well, this is 7 years ago, so maybe we should have an update and see how well things can be done today. And so actually there was another biocreative workshop just last November. And there was not exactly the same not exactly the same task, but a similar one. That was about drug-gene relation type extraction. And the best performing system had an F-score of just below 80%. So that sounds a bit better, but actually it isn't, because it's just the relation type extraction. So it's if you already know A and know B, to figure out what kind of relation there is between the two, right? So how can we also get A and B? They didn't do it in this task, but there was another one. And this involves named entity recognition. So recognizing that a certain stretch of text denotes a thing, in this case a chemical, and then link it to the correct formal identifier. And here the best systems, again, world-leading teams working probably months on this, they achieved F-scores between 81 and 87 for these two tasks. And you have to do both together, right? So with a quick back on, of the envelope calculation, if we have a scientific text that says something like thing A has relation R to thing B, we have to first recognize and link A, then the same for the thing B, and we have roughly the F-scores we can expect for this, and then determine the relation R. And errors accumulate, so we can roughly just take the product of all that. We can expect an F-score very, very roughly of around 40% to get a full relation out. So that means, with such a system, around 60% of the retrieved relations will be wrong, and 60% of the relations that are in there we're not going to get out. So I think it's obvious for the majority of possible applications we could do with that, this is not even far from good enough, right? So text mining is not good enough, and we need better text mining. Or do we? Frank has already pointed to that problem, that we should maybe see it differently. And I'm going to have the same quote again by Baron, who will speak just after me. Maybe there's a problem with text mining, because we only need text mining because we bury it in the first place. The researchers presumably knew what they wanted to write down, what it meant, but then they buried it in text. And then text mining has to do this task at which it's not very good at, actually. So can we publish without burying? That's what we wanted to find out. And we've made a first step, a kind of a field study, as we call it. And I think we succeeded, and we'll tell you more about that. We wanted to have a special issue at our journal, Data Science, that only has papers that do not bury their results. So that means that the main findings of these papers should be in a machine interpretable manner, should be directly accessible to software, to machines, to algorithms, whatever you call them. But there's a first kind of even theoretical problem. So how do we represent main findings of such papers in a machine interpretable manner? Because we kind of had the feeling, and we wanted to find out, that such results often, such findings, are not just as simple as A has relation R to B, but that they probably often are a bit more complicated. We wanted to find out, and for that we selected 75 random articles across disciplines, and we manually looked at them and we tried to elicit the main claims of these 75 articles, and tried to figure out kind of, is there a logical pattern that we could come up with that would allow us to represent these. And we found indeed one way that seemed to work well for many of them, and we formalized that as the super pattern, and we'll just very quickly walk you through it. Let's take the same example from the article as before. So that's just the title. So we did not look only at the title. We kind of read the abstract and often the full paper to figure out what is the main claim. And then we figured out we could use this super pattern, that's the blue part, that consists of 5 slots to be filled. You see there are 3 class slots that take any class. So the context, subject, and object. I show here just the class name, but these are actually class identifiers. And then the qualifier is a qualifier like generally, which we define as in at least 90% of the cases, from a relatively small list of predefined entries. And the relation also from a smaller list of predefined entries with relations about causality and collocation, etc. I'll just very quickly paraphrase what this means in more or less plain language, so you get an idea. So we start at the subject class. This means whenever you have a thing that is of type transient oxidative stress, that is in the context of a thing that is of type red brain endothelial cell, then generally, meaning that in at least 90% of the cases, it has a relation of type effects to a thing of class PGP expression, so expression of that protein, in the same context, so in the same instance of a red brain endothelial cell. And we can express that as logic 2. I'll not go into details here at all, but just this is something that an algorithm can understand. It's perfectly, precisely defined what that means in logical terms. So we went back to these 75 random articles and the main findings we found therein, and tried to apply the super pattern that we found. And the good news is 68 out of these 75 could be represented with the super pattern. And the even better news is that the remaining 7, they were not too complicated, but rather too simple to be represented. It turned out these were the simple kind of A has relation R to B type of statement. So these are easy to represent, logically speaking. And as a small side note, the red ones, these are the ones where existing text mining, relation mining approaches get an F-score of 40%, right? We can expect, we don't know, we can expect that such approaches work even much worse on the blue ones. So now we have a way, we think, how we can represent scientific findings in a general manner. So now we can apply that to publishing and actually have a special issue of papers that do it in that way. Well, we made one practical concession to that. For that, we introduced the concept of formalization papers. So our special issue consisted of formalization papers, and these are papers whose novel contribution is not a finding itself, that existed already, but the formalization of the finding. So we see that as one step towards our vision where we publish in that way original finding. So if a researcher finds something new, they publish it in that way right away. Whereas now we took findings from existing publications and in such a formalization paper, only the formalization is new, not the finding itself. But the whole setup, all the concepts, all the tools, they work just as well we think for this case where the findings are original, but for practicality, we did it in this first special issue in this way. And these formalizations are represented as nanopublications. I'll just show you here one example as it shows up in the interface the authors that submitted to this special issue were given. So we used this nanobench tool that we developed, and here the blue part, you see the main part of the nanopublication, which consists exactly of the formalization with the super pattern that you've just seen. You see roughly that there are 5 drop-down menus that correspond to the 5 slots in the super pattern. I just quickly read how it is shown here. So you see another example. In the context of all things of type human, things of type STX1B mutation frequently have a relation of type A occurs with B to things of type epilepsy. And the authors of this formalization paper then informally give a label that says mutations in STX1B are associated with epilepsy. So that's already kind of the main content of the nanopublication, but there is a bit more. There is some metadata, most importantly the provenance that tells us how this assertion came about. And in this case, it tells us that there was a formalization activity that took as input an existing paper. So the assertion is a formalization of a claim that occurred in an earlier paper. Then in the end, the authors pressed publish there, and then there were a number of steps. We went through reviewing, so they had to submit it first, then we went through reviewing. We did that as nanopublications too, but I don't have time to show you details of that. But in the end, we accepted 15 such formalization papers, and it might be a bit too small for you to read, but I have a link to all the slides on the last slide, so if you are curious. And actually, these are all links, so you can actually click and see what identifiers are used. Every row here is one paper, and it summarizes the super pattern that was in that paper. So every row of this table can be translated to logic. So everything here is in the back, it's machine readable, and it's automatically interpretable. Well, if you go to the publisher's website of our journal and to the special issue, it will look like this. It looks actually as any other paper. So you don't see a lot of difference on first sight. There is a get PDF button. You can get a PDF. It has an abstract, so a human can read it and hopefully understand it. But there's also this. There's also this link to a nanopublication. So you can actually, when you click on that, you see the same thing, the same formalization paper, but in nanopublication format. So it will then look like this. And we don't want any human to necessarily look at that, but it's of course for the machines, for the algorithms that can actually understand this. So what we've done is we have this special issue where all these papers are published in such a way that we can directly, trivially get to this. So we don't need text mining. Or a different way to put it, text mining is trivial. It's just about downloading a file. And this is then at 100% F-score. So that's what we have done. And I believe with that, I would again like to welcome you to this new era of scientific publishing that I think we've entered in a bit. Hold on just a second, because I only want to stress this was not only my own work, but also, most importantly, Christina Buchers, who is the PhD student who was mainly in charge of this, all that I've presented, and my co-authors, Davide Geolin and Jaco van Ossenbrugge. I have the link to my slides as promised, and some other links here. And with that, thank you for your attention, and again, welcome to the new era of scientific publishing.